So good morning friends. Today we try to understand how the vertical gel electrophoresis will work. This is the vertical gel electrophoresis instrumentation, okay? And this gel electrophoresis is mainly used to separate the mixture of proteins. If you are having different proteins having different molecular weight and you try to separate, then you can use this gel electrophoresis. So this is very small unit, and this is very simple unit of gel electrophoresis. So here you can see the two different tanks are there. This is the upper tank, this is lower tank. Upper tank, they are having one electrode, this is negative electrode, black one, right? And this is called cathode. This is lower electrode having another electrode, and lower tank, this is another electrode, and this is the positive electrode or called anode, right? Another component of the vertical gel electrophoresis is the casting plate. Okay, so these are the casting plates. This is complete plate, this is notched plate, right? We set like this, and then we put spacers in between the plates, right? So these are the spacers, 3 spacers we put here in between the plates during the casting of gel. Okay, and this is the comb. We put comb here in the notched part of the plates. This is the spatula by which we remove the gel after running, and this is some small device by which you can clean the wells. If some stuck acrylamide is there, you can clean by this apparatus. This is the power pack, as we mentioned in the earlier video. Same power pack can be used here, and this is the cord by which you can run the gel. You pass the DC current, so this device will convert the AC current into the DC current. So positive and negative electrodes are there, so you can fit here and you can run the gel. How we should make the chemicals? So all the chemicals should be made in a double distilled or the milky water if possible. So almost 12-13 chemicals we have to prepare for the Sds-Page. First is the acrylamide, base acrylamide solution, which is approximately 29.2 gram of acrylamide and 0.8 gram of base acrylamide, and 100 ml distilled water has been mixed. And then second chemical is separating gel, which is tris, 18.15 gram, and this should be 1.5 molar and pH should be 8.8. okay, and this is for 50 ml I think. Yeah, you can calculate. Then another is the stacking gel buffer, which is 0.5 molar tris and pH 6.8. Then fourth chemical is SDS, which is 10%. You can make 10 ml, it is fair enough. Then next chemical is APS, which is 10% can be made, or you can add a pinch of SDS as it is. Then another one is the timid. Timid is ready-made available. Then another chemical which is staining solution. If we go for the Coomassie staining, then Coomassie blue, then methanol and glacial acetic acid as well as the water should be mixed in that proportion. Then next chemical is sample buffer. Sample buffer, many a times it is readily available, or you can also make it. This is 9th number is the sample buffer. So you can take tris, SDS, glycerol, beta-mercaptoethanol, bromophenol blue and distilled water in the given proportion. And next is the tank buffer. So tank buffer, this is you just take the tris, glycine as well as the 10% SDS in this proportion. Okay, and in addition to that you can make the bromophenol blue, right? Apart from that, we should make the destaining solution. Okay, so after staining, we destain the gel. So destaining solution, it contains acetic acid, then methanol and double distilled water in the given proportion. All right. So these are the chemicals which are made. Our chemicals are ready. Now we prepare separating as well as the stacking gel. So for making separating gel, we are making here 10% separating gel, right? And depending upon your protein, you can prepare or you can decide the percentage. but here we are making 10% separating gel. So in between the plates, first of all you cast separating gel, then after stacking gel, because stacking gel is always on the top of the gel. So for separating gel you mix 4 ml of double distilled water, then 30% acrylamide bisacrylamide solution, 3.33 ml, then trace buffer pH 8.8 having molarity 1.5 millimolar, right, that is 2.5 ml, then timid, this is 5 microliter, then SDS, this is 100 microliter, then APS, either you can make 10% APS, that is 50 microliter you have to add, otherwise you can add a pinch of the APS, okay, as we added in the video, we will add in the video. Then you make the stacking gel, and stacking gel, this is 4% or 5% depending upon your protein of interest. So double distilled water, this is 3 ml, then 30% acrylamide bisacrylamide 0.67 ml, then tris having pH 6.8, that is 1.25 ml, then timid 2.5 microliter, 10% SDS 25 microliter, and 10% APS 50 microliter, right? And then you cast accordingly. First you cast separating gel and then stacking. We have to first seal the spacer to the plates. So these are the spacers, you can see here, these are the spacers. So 3 spacers are there, right? This is 1 millimeter thick spacer. So we can use grease, otherwise we can also seal without grease, but it is always safer to have grease, right? So we put grease to the one surface first like this, right? So it will stick your grease will stick your spacer to the plate, okay, and the chances of leakage becomes less then. So like this we put first of all at the lower end, so just we kept here, right? Then we take another spacer, right? So another spacer, again we put, you can use Vaseline also, Vaseline is very easily available, Vaseline is nothing but the grease only, right? Otherwise you can put grease also, okay. So like this, right? So you can put this spacer at side, okay, we kept it here, and now we take third spacer. So again you put some grease over there, all right. Moderate amount of grease is all right, okay. So this is the third spacer, all right. And now you can put some amount of grease on the top of the spacer also, so that the other plate can fix easily, okay. See, I'm again mentioning here, there is no such thumb rule to put the grease, right? This is just to align your plates well as the chances of leakage also becomes less, right? But for making the gel leakage proof, we seal the gel with the agarose also, right? And now you put this notched plate over here, okay. So we put notched plate here, fix them well, right? Just you press them, okay, all right, okay. And what you do after that, just you take these clips, these are the simple clips, right? You can get these clips at the stationery store, okay, or scientific store, and you put these clips here like this, okay, so that it will be fixed well. So both plate is now fixed well, okay. like this. So now this plate is ready, but if you cast your gel in this plate, the chances of leakage is there, right? So to protect or prevent the leakage, we seal this with the 1% or 0.8% agarose, right? So this is the agarose. You just put agarose and then to the side of this one, see, like this. Okay, we'll take some more amount of agarose, right? Just you run the 1% agarose, it will become seal automatically. See, like this. See, this is the agarose and this is just sealing, right? So we sealed. This is almost sealed. See, now you can check by putting water in this, in between the plate also, and you can check the leakage, right? But if you are confident, then there is no need to check the leakage. So now our plate is ready. So now we have to cast the gel, right? So for casting gel, we have two different kind of gels here. One is separating and another is stacking gel. So first of all, we should cast the separating gel, right? So for separating gel, we have to take distilled water first. This is 4 ml. For 10 ml gel, we are preparing 10 ml of the separating gel, right? So we are taking 4 ml of distilled water. 1, 2, 4. So this is our distilled water. Then we have to put 30% bis-acrylamide and acrylamide solution, which is 3.33 ml, right? We are making 10% acrylamide gel, all right? So for 10%, this table is there, what we have seen earlier, right? So now we have to put 3.3. So first I am taking 3 ml. See, now the acrylamide and bis-acrylamide solution is neurotoxic, so take care of that. You should wear the gloves, and if gloves is not there, then at least you should put the mask. Don't take or don't do mouth pipetting, right? So these are the cares that should be taken. So this is 3 ml. Now this is 3.3, right? So just I am setting the pipette. So this is 3.33. So now this is 3.33. All right. So this is, and mix well. See, mix the water and the acrylamide bis-acrylamide solution well. Okay, then we have to take 4x, tris buffer, which is pH 8.8, right? 1.5 millimolar, right? That is called separating buffer, right? So acrylamide, this is done. So you cap it. You always keep your chemicals in front of you so that you can separate gel. Okay, now this is the separating buffer. So we have to add 2.5 ml separating buffer. So I am adding first of all 0.5 and then we add 2 ml. So this is 0.5. So this is 1.5 molar pH 8.8 separating buffer. So this is 0.5 and now we put 2 more ml. This is cocktail for the 10 ml, right? 1, mix it. Always you keep on mixing. Okay, so now this is 2.5 ml. So separating buffer 2.5 ml, 1.5 millimolar pH 8.8 has to be added. Okay, so now the timid, another chemical which is called timid. So 5 microliter timid should be added. So 5 microliter is very less amount, so you have to take the appropriate pipette. Okay, and smell of this chemical, the smell of this timid is very unpleasant. So this tip would be all right for this pipette. Okay, yeah. Then we have to take timid. See, this is the timid. Okay, so open it and just 0.5 of 5 microliter. Oh, the tip is gone inside. Okay, and you put it inside and then you press the pipette and mix it well. Okay, so this is timid. So timid has been added. Keep it properly. And then another chemical, because we are preparing the Sds-Page, so another chemical is Sds, and here we have to mix 100 microliter of 10% Sds, right? So I'm mixing 100 microliter. Okay, no problem. We set this at 100 microliter. So this is 100 microliter, and this is SDS, 10% SDS. This is already prepared. All right, this is 10% SDS. See, so 100 microliter we have to add. So added, and then you mix it properly. Always mixing is always very important, right? So mix it. See, now the last chemical, which is APS, right? So APS is very important chemical here, and once the polymerization started, when you add APS, so see, APS you can add a pinch of SDS into it, or otherwise you can add 50 microliter APS, 10% APS. You can prepare solution also, but here I am adding the pinch of SDS, this APS. So just you take a pinch of APS here. I am taking. Some more it is needed. It is too much, and just you see, for 10 ml just a pinch of APS, and fresh APS it is required, right? So don't take some old APS or like that. So you can add, and as you added, see, as I added this, so polymerization started now, right? So now you should do the things very quickly. You should pour it now, right? So we have to pour 2/3 approximately, right? So 2/3 of the plate, right? From the nost plate you have to take. So up to here we have to pour, right? So you pour it quickly now with the help of pipette, or you can pour it like this also. Depends upon you how much practice you are having. See, now you put it like this, and little bit also we have to add. Okay, so we add it. Okay, and always take care that bubble should not come, right? Okay, all right. So this is separating gel, and if needed you can add some water layer on that, or a butanol layer on that, over that. Otherwise, I mean, it is not the necessary chemical to add. All right, and then after 15-20 minutes we then make the stacking gel, right? You can see we added water over there, separating gel, so you can see the line, a very thin line is there, and this indicates the polymerization. And why water we added? Because water it breaks the contact of gel from the oxygen or air, right? That's why it polymerizes. So now what we do, we just discard the water. See, we discarded water, right? Water you can add also butanol, right? Depends, right? Depending upon your requirement and what chemical you have. So water is available everywhere, right? So you use water, right? So now you can see, so our separating gel is ready right here. Okay, so now we cast stacking gel, right? So for stacking gel the chemicals are almost same, okay, concentration and amount would be different, and just the stacking buffer, only the additional, it is not additional, the separating buffer is replaced by the stacking buffer, right? This is 0.5 molar and 6.8 pH. Okay, so so now we cast stacking gel. Okay, so for stacking gel the chemicals are double distilled water 3 ml. Again we are making approximately 5 ml, right? 5, 6, 7, right? So this is 5 ml of stacking gel, right? So this is double distilled water, so 5 ml double distilled water is required for that. You can see the table, right, what I provided you. So This is 1, 2, 3. All right. So 3 ml double distilled water or milky water you take, then 30% acrylamide, 0.67 ml. Again you refer the table, right? Then 0.67. So I'm setting here 0.67 ml. Right. So this is 0.67. Okay. So we used water, so we can use the same tip here. So this is acrylamide and bisacrylamide solution. So you add and mix it properly again, right? So mixing is very important here. So mixed it and then discard the tip. So this is acrylamide, and then the third component is tris, right? So this is the tris, I mean stacking buffer, right? 0.5 molar tris, pH 6.8, and this is 1.25 ml. So first of all I'm adding 0.25 and then 0.25, right? Always mix it properly, then 1 ml. Yeah. Mix it well, mix it properly. Then the fourth solution is timid, right? So timid here 2.5 microliter we have to add. Okay, so this is 2.5 microliter. So just you take timid. Okay, so mix it properly. Okay, and then next chemical is 10% SDS, and this is 25 microliter, right? So we have to add 25 microliter here, SDS, 10% SDS we already made. Okay, so this is SDS. Yes, 25 microliter. Mix it properly. Okay, last chemical, this is APS. So mixed it properly, everything should be ready before adding the APS. See, this is 1 millimeter thickness comb, so we should place the comb like this here, right? But before that the comb should be ready, right? So comb is there with us. Now this is the APS, right? So just you have to add APS, a pinch of APS, right? APS. Take APS now. So see, this is fair enough. Okay, so just you cap it first, otherwise it will get oxidized. Then you mix it, right? So mix it properly and do the things very quickly, right? Otherwise it will polymerize in the beaker itself, right? So we mixed it, right? Now you pour. You can pour with the pipette also. If you are not, having trained hand. So see, till the edge we filled, and now our comb is ready, right? So now we put the comb slowly here, right? Slowly, just I'm putting the comb here, right? So it should be straight, so that you keep in the mind, right? Yeah, this is fair enough. Okay, let me take a little more. Okay, so now our separating gel as well as the stacking gel both are ready, right? And now we leave it for 10 minutes. See, now our gel is ready, right? So now slowly we remove the comb first. So I'm removing here comb. So slowly you should remove, right? So I'm removing very slowly. slowly you have to move. Okay, so it is now well is ready, our well is ready. So now you clean the wells first, right? By having the distilled water, you should clean the wells very much clear. It should be, your wells should be very much clear, otherwise during the loading of your sample you will face many more problems, right? So just by having the distilled water, I am washing the wells, right? So you can put it here, no problem. See, this device is for making the wells clear, right? If something is stuck onto the, like, see, this is the gel which is already stuck to the glass plate, right? So you can clean, otherwise you will get some problem during the loading of your gel, loading of your samples, right? So you can clean it like this, okay? And make sure that your well is very much clear. You can clean again. I can see it needs a bit more cleaning, because some of the, like this needle, this simple needle, you can clean if something is stuck into your well. See, okay, so nothing is there inside. Whatever you can see, this is just on the outside of the well, okay? So now this is clear, okay? You can wash it again if it requires. Okay, all right. Okay, so now what you do, you just remove the clamps. I'm removing the clamps, right? And after removing the clamp, you should remove the lower spacer, so that it will, or current will flow, or current will make a circuit, right? So this is the lower spacer, right? And now you put the gels, notch should be towards the well, this tank. So this is the small tank, so I'm fitting like this, right? ये इस तरह से fit करना है. Then you tighten it, right? So I'm tightening. So now this is just little bit, so that it will make the circle, current circuit should complete. Tight it properly, otherwise your buffer will leak, right? Okay, and now we have tank buffer, right? Now electrodes are here. Now you add your tank buffer, and this is our tank buffer. So I put my tank buffer here in the upper tank, and this is in the lower tank, right? Yeah, okay, that's it. Okay, so see, now I put the electrodes, or we started the current, right? And now I am loading the samples, right? So sample is prepared. You just go to the protocol and see how to prepare the samples, right? So sample is prepared. So now I will load 50 microliter of sample, right? So whatever sample we prepared, I load only 50 microliter first, right? So now you put like this, okay, and slowly you press the pipette, okay, slowly, so that it can settle down. is available. Hamilton is a kind of pipette, micropipette, by which you can load. In that case your sample won't spread. Here the samples are spreading actually. Okay, no problem. So this is one sample, right? I am loading other sample. See, this is nice actually. See, by this tip I can load very smoothly, but the problem is that you can load only 10 microliter in one pipette or one shot, right? See, only 10 microliter I am loading here, but best thing is that your sample won't get spread, right? Yeah, all the samples has been loaded, so we put them now onto the 125 volt and 43 milliampere current, and will run for maybe 45 minutes. Okay, so now our gel is almost completed. Run is completed. You can see the tracking die, it comes almost the lower level of the plate. So now I switch off the current, I remove the plugs from the electrodes, okay, and now I am removing the gel. Okay, so just you open like this, and see this opening, it depends upon the apparatus to apparatus. Okay, so here this facility is there. This is very small gel actually. This can be called as baby gel. Okay, so yeah, so now I removed. Okay, so now you can see the status of the tracking die. Okay, and now I just remove the spacers very carefully so that it won't break the gel. Okay, so I removed one spacer. Okay, then I am removing the another spacer. Okay, and with the help of this apparatus, this is just the gel remover actually. So I'm removing the plates. Okay, so slowly you can make some pressure, or you can just push inside. Okay, so now this plate, so now be careful so that your gel won't get break. So now this is stuck to this gel. Okay, so I have removed the bigger plate or the complete plate, and now what you do, you just take the staining solution. So this is the staining solution, right? And you put the staining solution inside any such box. Okay, so now I am putting this staining solution, and before that you can wash also, right? So I need one more plate so that I can wash it, right? Otherwise I can wash. Okay, so now you can see this tray is there, and you just put some water, distilled water simply, or double distilled water, right? You just put distilled water. This is just the washing process, nothing more than that. Okay, so I washed it from upper side, right? And now with the help of this spoon like apparatus, just you remove the gel very carefully so that it won't break. Okay, all right, so now I'm putting some water here, right? And see, now our gel is there in the tray, right? So now almost the washing process is completed. This is the washing, right? And now I transfer this gel into the staining solution and will keep it for overnight. Okay, so this is our staining solution, and now you can you can hold this gel, because this gel is bit stronger than that of the this agarose gel. So it is not a very brittle gel. Not this is brittle, but this is not so brittle, right? And now I hold like this, and you can put it here, right? So now this gel is in the staining solution. So now I keep this box along with the gel in the staining solution on the rocker for overnight, right? And then tomorrow we destain so that we can see the bends. So now you can use, or one can use this buffer, tank buffer, 2 or 3 times, right? So I'm just preserving this buffer, right? So you can just pour this buffer carefully into some apparatus, whatever you are having, right? so that you can use it again and again. I mean, at least 2 times we can use, right? 2 times we can use very easily this buffer. Okay, so just like that here. Now this is the gel after overnight incubation in the staining solution. So now I am pouring down the staining solution, right? And then we wash it with the simple double distilled water, and then we put destaining solution, right? So just I am opening. So this is the staining solution. So staining solution is coming out. And see, even though in the presence of staining solution, if your protein concentration is too high, so in that case you can see the bands. If your protein concentration is too high, very high, right, in that case you can see, right? And this staining solution can be used, right? This can be used at least 3-4 times. So don't discard the staining solution, right? Staining as well as destaining, these solutions can be used. So this is just simple water, right? I'm just rinsing the gel with the staining distilled water. See, if you can see carefully, you can see the dark spots are there on the gel, very light, because the background is blue, right? So you can see here the dark background. So these are the protein bands actually, right? After destaining, you can see these bands very easily. So just I'm tightening this notch, and I'm putting the destaining solution here, right? So this is our destaining solution. We made destaining solution. This is nothing but the acetic acid, methanol and water, right, in the given proportion. Okay, so this is destaining solution, right? Okay, yeah. So now you incubate again put on the rocker for 1 hour or 2 hours. Then after you change the destaining solution again. See, now this is the gel which is kept in the destaining solution, and we kept for approximately 2 hours, and after 2 hours you can see the status of the gel. So just I am putting it out. Okay, and we can also pour carefully like this. Okay. So see, here you can see, if I keep this gel like this, see here you can see the wells. I'm putting it. So these are the different bands. Again we have to destain. At least 2 to 3 times we can destain. Okay. So these dark bands like this band, these are the overexpressed protein actually, right? And here is the marker, right? This line is the marker. So you just do 1 or 2 more destaining, and you will get very clear cut gel. Okay.